Well, maybe I could get you guys to gather up a little bit closer. We're just going to spend about 5 minutes introducing the next module, and then we'll be splitting up to groups, doing a little bit more interactive stuff. So, what do we call this module anyways? Basically, it's precision ag tools for on-farm research, and we're pretty excited about working in this area. First of all, I'd like to introduce the instructors. Louis Baarda is our newest employee with Farming Smarter. He's got a master's degree in GIS, so he's somebody very important to me when it comes to dealing with the data associated with precision agriculture. So I started it off usually as that no data is better than bad data. And it was actually Bob Blackshaw. He's been telling me that for years, and I've really grabbed onto that. That's a real big problem with precision agriculture in general, is it's really easy to create bad data. And when you create it yourself, you've got a bias there. Like, there's so many biases involved that we don't really fully understand. But the good news is that the scientific method was actually designed to help us be aware of bias, to control it, to understand it, and deal with it. So the module today is really about understanding bias, understanding error, and how do we apply the scientific method to do effective on-farm research. Because if you're going to go out and create out a bunch of junk data, don't bother. Because there's a tremendous investment in on-farm research. We all know that. That's why it hasn't really exploded or taken off yet. Because there's time involved, there's money. Yeah, sure, we all have GPS. Yeah, a lot of us have yield monitors. But what is it that we can generate data up the yin-yang, but how do we convert that into knowledge that is both practical and applicable to your farm so that you can make money? At the same time, I have seen it done properly, and I've seen guys embrace it. It's like that first time you walk into a casino and you pull the one-armed bandit in your first draw and you win $1,000. You're hooked, even though you may lose $5,000 the next time you go in there. Lots of situations I've seen, farmers can make an additional $20,000 on a quarter for what they've learned through on-farm research. And now they're managing their entire farm with proper rotations. Now, I'm not suggesting that we all need to do that, but at the same time, I can tell you that unbiased data information that we're all looking for, it's getting harder to find. Governments that we used to rely on for that over the last number of years, they're getting out of the business of it. And more, we're going to have to rely on ourselves for creating our own information. And organizations like ours, but we can't cover it all. I mean, there's just so much out there in agriculture. It's such a complex industry. So our goals, first of all, we don't know it all. We don't know even close of it, but we have been involved in a number of projects. The first project right now that I just wanted to mention briefly is, and it's using these instruments off to our right here. It's about studying the variable rate side of things. I don't want to confuse you with variable rate, because today we're talking about on-farm research. Variable rate is just one component of on-farm research. So the instrument there is a Varus. It's a soil mapping device. That's one of the biggest challenges. You've seen some of our small plots. Small plots, we do that for a reason, because we want to be able to control the variation and have confidence that what we're doing as a management practice is literally what's creating that effect and not the variability in soil. So we pick nice uniform areas and we do things small, and we understand the error throughout of all of our equipment, from our small seeders to our plot combines. Everything we do, we try to minimize that error so that we can say with confidence at the end, yes, this happened or this didn't. So that machine there, it's an electroconductivity mapper. There's certain consultants out there that are employing them for mapping soils. That particular one's a newer model. It actually maps pH, electroconductivity, and organic matter. In addition to that, we're pulling behind it, you'll see in a little calf sled, an EM38. That's another type of electroconductivity sensor. So we're mapping fields, and then we're using prescription on-farm research to validate whether there's a response curve associated with the differences these maps show. A second project that we're working on is using multiple years of yield maps as a way to just do variable rate research in canola. That's the first year so that we're doing that, but we're using little study blocks within a field. Second introduction is Dr. Adil Akbar. He has a lot of experience in the spatial side of things, so understanding spatial variability, statistics, that sort of thing. We're going to split up into two groups. The first group is going to head over there with Joel Huber and Blaine Metzger. They're with the Ag Tech Center, Alberta government, and we're going to talk about if you're going to embark in this scary, wonderful world of on-farm research, you're now using your seeder, your combine, that's your tools for research. So you need to understand what the limitations are. You know, what are the errors associated with that? And in knowing that, that's going to decide on what types of research questions are actually legitimate in embarking on. There's no point in trying to pick out, 1 or 2 bushel differences when you have 10 or 15% error. Second group, you're going to split up with us, myself and Lewis and Adil, and we've got some virtual fields. You get to play the almighty God farmer, essentially, looking down on your fields, and we're going to go through the steps of creating an on-farm trial. We've got some scenarios, and it's going to be a little bit of interaction there.
So at the end of the day, you can pare down field scale research to about 4 different steps. So the first is, what is your research question? Then to choose a design that's going to help you flush out the solution to your research question. The third is implementing your trial. And then finally, your data collection and your analysis. So I'll just do a quick hit on each of them, and then we'll split up. So as far as the research question is concerned, it seems like a simple thing to say exactly what is it I'm trying to figure out, but it can be a little bit tough sometimes. And I think the biggest thing to think about there is to keep it simple. When you're looking at a full field scale trial, there's a lot of variability that whether it's weather or in your field, if you have a complicated research question, it can be really tough to flush out exactly what your results are. So you want to keep it simple, keep it to a yes and no question, or A versus B, or A versus B or C. But anything more complicated, if you're trying to do 2 factors at once, say seed varieties as well as seed densities as well as nitrogen levels, the more you introduce, the tougher it is to kind of keep your results rigorous. Yeah, and be specific with your questions. So know exactly what your outcome is and what you're looking for. The second step is looking at choosing your research design. And again, field scale research, the biggest thing we're trying to do is trying to eliminate any variability. So eliminate any external effects that might influence the results we're looking for. So if we're trying to determine, seed variety A versus seed variety B, we want to be certain that the differences that we see at the end of our trial isn't a result of soil conditions, or it's not a result of weather conditions that they may respond to a little bit differently. So there's 2 things we do to try to mitigate those effects. The first is to replicate our trials. So in your fields, to set up a trial where half the field is one variety and half is another variety, it's really not the most effective way to do on-farm research, because we cannot be sure that the effects, that the results, the yield results at the end of the trial are a result of the treatments themselves, the different varieties. It may be wind effects on one side of the field affecting the one variety more than the other, or weather effects or soil effects. So when we replicate it, if we do three different sections for each of the treatments, then it gives a little more rigor to our results. We know that the results we see aren't a result of natural variability, whether it's in the field, in the soil, in the weather, or even in a plugged nozzle on your spray or something like that happens halfway through. So that adds a little bit more rigor. And the second part of that is to randomize the location of your treatments within your replications. If you have the same seed variety on the west side every time, there may be, in southern Alberta, there may be a wind effect on that, for example, where whatever's on the west side gets dried out a little faster. So at the end of your trial, you're going to feel strongly that one variety is better than the other, but it may have nothing to do with the varieties themselves. It may have to do with a wind effect. So by randomizing the locations of them, so once one variety on the west and then the next on the west and then the first on the west and the next on the west. So replication and randomization are 2 things to really keep in mind, and that's in our small plot research too. That's kind of the foundation of what we do to mitigate the effects of variability. So number 3 would be implementation. There's a few things to consider there. And Ken talked a little bit about precision agriculture. And it's important to keep in mind that precision agriculture it's a tool, and it's something to kind of help you out with it, but it's not the be all and end all. You still need to set up a good design and to consider how to use that to make your study successful. So for example, one thing we've done with some of our trials is to try to line up our seeders and our combines together. So if we have a 40-foot seeder, for example, and a 30-foot combine, we'll do our treatments will be 3 passes with the seeder, which will line up with 4 passes with the combine. And that way we don't have any overlap between our treatments. So that's, as far as implementation is concerned, that's definitely one to keep in mind. You may want to consider leaving the edges of the treatments and harvesting them separate. So when you harvest each of the seed varieties that you're really looking at, you want to be right in the middle of that. You want to make sure that you're getting the information that you need, not a combination of 2 different treatments together, because that'll throw off results as well. And record everything. Every practice, everything you do to that field over the course of the season, you want to know what you've done to it, whether it's the amount of nitrogen you've put in, what you've sprayed with, what time of day you sprayed with, what kind of weather you had. And then in hindsight, or if you do the same trial over a couple of years, you'll kind of have a bit of an idea what the effect of those were on your results as well. And treat everything equally. So if you spray, whatever you spray, well, spraying is a good example because you want to make sure the effects of the tires are the same for each variety you're looking at. So if you have 2 tire tracks in one treatment, you want to make sure there's 2 in the next, so that whatever effect that has on your yield results, that's equal for each treatment. And the last thing is data collection. You want to make sure that you're pretty rigorous with your data collection, that you treat, again, everything the same. Consider calibrating your equipment, maybe use the same combine for the whole field, because the yield monitor in combine A may be a little bit different than B. We typically cut out the headlands. There's a little bit different growth responses in the headlands. Due to just a lot more driving there, or being on the edge of the field, wind effects again. And the final thing is, as far as analysis goes, is to consider using statistics. I think, given things the old eye test, or just a basic this yield is half a bushel more than this yield. In the grand scheme of things, a half a bushel might not be a really big difference. So that's just a quick hit of some of the basics for on-farm research. I don't know if Ken or Adil have anything to add there. Yeah.
I think the biggest thing too is that in your manuals on page 26, what we've done is try to keep it as simple as possible, but to include all of the key steps that we think are important in precision agriculture. And we have somewhat distracting, but still a pretty cool demonstration of an octocopter going on right now. There's been a lot of interest in, this is a perfect example of a technology that is available, but yet we still have to figure out how to implement it effectively so that it's not just some big kid's toy. And precision agriculture is the same thing, right? We've had precision agriculture for 20 years. There's tremendous technology, but we need to put together the right skill set, the right motivation, so that we can use these tools effectively, so that they're valuable. And we're going to be working with some of the remote sensing type things, and I think there is potential, but there's also a real need to delve into the information that is generated from it and try to make it practical. So page 26, what Lewis sort of touched on too, that I think about too, is most of us and most of people in the farm world, they're not really experienced in research, they're not experienced in data analysis, but as an organization, we're trying to build capacity so that we can help out with that. I'd love for us to play a role in helping coordinate, helping advise on-farm trials. So if you're at all interested, certainly give us a shout. There are other people that are involved there. There's some companies too, but there's more and more interest in getting sort of third-party academic type involvement into precision agriculture. So on the technical side of things, because it is difficult, and the work that Adil and Lewis do in analyzing the data and pulling out the spatial features that are influencing results, that's not really the general farmer type type application. In fact, a lot of the software that's available for precision agriculture to farmers is very limiting in what you can do with the data. And for that reason, the better planned setup that we can in producing clean data, the less post-data analysis that needs to be done. And that happens to not be any of our strong points. So if we set it up properly, then you don't have to do so much of that post-analysis, but it is available. We're going to try to put some more proposals together that are going to come up with some free and some easy to use applications that will allow you to run statistics on some of your trial results. There's also a tremendous opportunity for collaboration. Once you're trained up and doing on-farm research, if you've got a few neighbors that are really big into it, you guys can form your own little research networks. You might be able to come to conclusions a lot faster and have a lot more confidence in your information. So I don't think we're there yet, but that's sort of our vision moving forward, that we can get to that point. Sound good? Okay, so a third of you now are going to head down to our virtual fields, middle part here and another third over here. Okay, let's take a look at this field. So this is your field now. Each one of you, whether you're a farmer or not, are now farmers. So
That's their best return on investment.
sell it. So our scenario here is that the wheat fields that you've been getting off this field have been consistently lower than your neighbor who's been growing the same variety as you for the last 2 years. Your neighbor suggests that maybe using a different seeding density will help increase your yields, but you think lower yields may be due to the quality of your soil. So one of the biggest issues in that type of a scenario is, I mean, there's already biases, right? Like you're like, oh, I got to compete with my neighbor, that sort of thing. You got to sort of forget about that. What is your true objective? You want to, in this type of scenario, you know that your neighbor happens to be getting a higher yield, but what you don't know is he really getting is he doing everything the same? Is his field the same? You don't know if this field is optimized, or you're just recognizing a difference that happens to be there for other reasons, right? So you're farming. What in this type of scenario, what do you want to learn? Like, what's your research question?
I guess I don't know if I'd want to see how to maximize yield, how to maximize profitability.
Well, that's perfect. So, whoops, sorry, Brian. I clocked you there. So that's sort of what I consider an objective, right? It's not necessarily the research question, but it's an objective. You want to maximize yields, but really what you want is maximum economic yields, right? So we'll call that optimum yields. So then in this situation, I'm gonna get you to remove that clamp actually, because I think it's fogging up pretty fast here. Just get a little air in there. There we go. So what factors, say, in an on-farm situation can we study then to determine whether you're hitting your optimum yields in this situation?
Seed density A versus seed density B. What
Okay.
you're doing currently versus what your neighbor's doing.
Okay, that's actually what comes up often is, so we'll call that normal, and what your neighbor is doing. Is that enough? Is that going to help you answer that question? And I think your question really is what is your optimum seed density, right? That's why it's important when we're rather than jumping into it, and that's instinctive, and I don't blame you for it, but I think it's wrong. The reason I think it's wrong is because what if your neighbor isn't at optimum and you're not at optimum, right? You're making assumptions that will lead you to improper conclusions. So what's a better solution? Anybody?
What you're doing now versus a few other options, not
Okay.
just one other option, but if you're doing 50, then try 100. maybe that's a little high. But
So what you're saying is normal, we'll say high, and low. That's not a bad solution. As long, see, what we do in that type of situation then is the data that you'll create, we'll say that's yield, and we'll say that's seeding rate, is you're going to create a response curve. You can do that with 3 data points, but you can't do it with 2. So 3 is probably the absolute minimum. If we did it this way, and you were confident that you were somewhere near that optimum isn't always the highest, but you can take all of your input costs and your yields, and you can plot that, and what you'll end up doing is finding the exact spot after the fact where your optimum was. So as long as you're getting that curve somewhere where that optimum is going to hit it, that's great. If you're really like new to it and you have no idea in a different situation, you might need a little bit more than 3. So more of a range. You know, you don't want to go down to a 0 seeding rate in this case because you'll have no crop. It's not practical. It's kind of stupid. In a fertility situation, sometimes you would, even if it's a small area. We do a lot of fertility research because it's really that crop input that tends to influence the most. But you also want to think about picking up big differences, because if you haven't been over there yet, you don't have that handle on the error. If you can't pick up big differences, then there's no point in doing that study either, right? So let's go with that. Do you guys want? We'll keep it simple. We'll just do seeding rates. But we actually had one group that thought, well, you know, I'm looking here. I've got salinity. I'm interested in seeding rates, but I'm also interested in cultivars. Maybe there's cultivars that have different salt tolerance. So how are you guys feeling? Are you feeling brave? Do you want to really get complicated, or should I stick to keeping it simple? Okay.
But now are we basing this on dry land or aerated?
This is dry land. Yeah. We have a pivot down that way if you want. We can go check it out. Yeah.
What about mortality rates in the salinity?
Like whether anything grows at all?
Yeah, well, like I'm from around Edmonton.
Okay.
We've had some guys double their seeding rates in their gumbo spots, and
Yep.
they still have the same. The mortality rate's twice as high there. So
Absolutely.
they get a stand after that.
So then when you bring that out there, that ends up being an objective, but are we answering it from this question?
Yeah.
Yeah, I think we are.
If you focus,
As long as you have that
part of
data
in the
collection
salinity
portion.
Zone 2, right?
Yeah. Well, you're touching on a very nice point here. So let's talk about it some more.
You can't do 50 plants there and 100 over here and say that 100's that much.
You say you want to keep it simple, just seeding rates? Chicken shit?
well, you don't want to do too much, Ken, because then it gets complicated.
You know what? We successfully did it last time. That's why I'm only asking on one of the groups.
Let's do it.
Okay, let's do it. Let's get complicated.
There's a little bit of knowledge here.
Okay. So yeah, we do have a little knowledge here. Are you afraid?
No.
Okay. So what we want to do in this situation is now we have 3 seeding rates and 2 varieties. So I'll call those factors. Can somebody give me a different pen? You should write it.
Me?
That's probably better.
Yeah. I don't need to be working anymore. Somebody holds that and you write it.
Write it on here?
Let's write that up there.
Higher? You got her.
Honestly, one of the messages I want you to take home is simple is important. But this is totally doable, and it's actually not as complicated as you think it is.
Doesn't work.
Where did...
That's litter.
It's college land. Yeah.
Fertilizer.
I didn't even ask them if I could use this area. So factor 1, seeding rate and seed density. So basically that ends up becoming what you call a factorial arrangement. You want to have all 3 of those seeding rates with the 2 different varieties, right? So you've got 2 factors. What ends up happening then to get every arrangement is you'll have 6 treatments in your trial, okay? So 6 treatments. Man, this pen sucks down here. So we've got we'll say variety X low, extra large, X normal, X high, and then we'll have Y low, Y normal. Can you write that on there too? And then Y high. Okay. So you have 6 treatments. We can number them. 1, 2, 3, 4, 5. What the hell did I do there?
You bunched 1 and 2.
Did I?
Oh, you see it from the side.
4, 5.
Just move your number 5 down.
Okay, 5, 6. All right, so we kind of have our objectives. We have our research question. We now have our treatment list. This is a treatment list. So let's think about the spatial arrangement of this study now, because what field scale research ends up being a challenge for is because of the field variability. And in this field, you know, we've got sort of emphasized variability. But what features do we have? We have the salinity zone. Do we want to? Should we be putting our trial in there?
I think you need to replicate it in all your zones.
Okay. You know what? My first instinct was, hey, this salinity sucks. I'm not going to touch this area. But in this particular research question, it's almost integral to the question. If it was another study, if I was looking at something totally different, there might be a situation where I would say it really doesn't make sense to put that study in that zone, because the variability there now becomes a confounding factor. A confounding factor is something that you don't know whether the effect was because of one thing or another. So we'll keep that in. But this part up here, you know, there's a haystack up here, and you know what? Farmer's been putting haystacks there for the last 10 years. So my suggestion then is let's just avoid this area right here. You don't have to use the entire field to do field scale research. You talked about whether our rocks were to scale. You know what? I don't think there's any point in going there. So that's okay.
I don't think there's any
Right?
good farm in there.
Yeah, so really what we are is we know that there's extreme variability. Yeah, there's only 4 passes anyways, right?
And half of it's gumbo.
So then in this type of situation, I would probably suggest let's just use this part of the field. So Brian, we can do, with guidance, we can just do entire strips all the way across, like that. Or we can go in this direction. What do you think we should do? Anybody?
Probably this way.
I'd go this way.
Hey!
Variability across all the.
I used to have prizes to give out, but yeah, I think that's exactly right. You want to explain?
Well, because if you run it this way, you can get the slope, the salinity, and the top and the
Yeah.
you can get it all running this way.
That's right. So what that's going to allow us to do is block our treatments.
Yeah.
And what everybody else does is, oh yeah, I'll do 1 pass of this and 1 pass of that. I'm going to tell you that's just totally inappropriate unless you have it in a block design. So randomized block design is one that we use in small plot a lot, and I think all we need to do is use that same methodology in field scale. So then let's block it. We'll do 3 replicates. A block is basically the same as a replicate, but what it does is it allows you to compare the differences from the average, and then you'll know whether there was both a block effect and a treatment effect. So in this case, we're very interested in this salinity area. It becomes a block. So why don't you let's draw 6 lines right here. That'll be our treatments. And the lines are kind of neat because it ends up being, you know, 1 pass of the cedar. In this case, 1 pass of the cedar actually lines up with this combine, this toy combine, perfectly. 2 passes of that combine is 1 pass of the cedar. You know what? I'm sorry, sir, I don't know your name. Don, do you want to draw 6 lines down there too? Maybe give him your pen and hold his papers.
Okay.
We'll try and learn.
Just get comfortable.
Yeah.
Yeah, do 6. Let's do 6 right here too.
So then you'll have 3 blocks.
Yeah.
Low, medium, high is the first one.
Then you'll have 18 data points instead of 6. Sorry.
Well, you'll have 6 treatments, but you'll have 18 plots essentially.
Space
Yeah.
is pretty good to also.
Yeah, ideally they line up here, but
We'll say that you just use an outback.
you're not a professional, I know.
Water's low.
Okay. So it seems complicated, but it's not. Okay. So now these blocks here, we don't have to treat them anything differently. We just seed all the way across. And what we do is we create a prescription for your seeder so that it's automatically going to change based on GPS coordinates. If this is a treatment, once it reaches this point, automatically change. And these prescriptions are not hard to make. The companies will often make it seem fancy, but it's not. You know, with some basic software, I've seen a guy put together a trial like this in 10 minutes. And then it doesn't matter. I mean, there are complications, technically speaking, in every monitor seems to have a different file format. But if you get to know your own equipment, it won't be that hard. For us, yeah, it was a challenge because one monitor takes this format and one takes a USB and one takes a flash card. That side of it's a pain in the ass, I'm not going to lie. But if you're dealing with your own equipment or with a set number of clients, you can get that figured out pretty well, which is an easier pen to see here. Okay. So then we'll call this our rep 1. We'll do it backwards. Rep 2, rep 3. So now we have our 6 treatments. I'm happy to share a little Excel add-on that we do that will randomly generate these trials for you. Totally free of charge and super easy to use. But I can use my brain random generator, or you can flip a coin for it. So now let's put our treatments in here. This is the randomization part. So I'll just use the treatment numbers because then it's a lot easier, right? So
How do you change cultivars on the go, though? That's the part.
we'll check out our seeder. How many tanks do we have?
3.
We got 3 tanks. We can do it with this implement. But that's a very good question. If you don't have the ability to do that, maybe we go to a simpler design like we were originally doing.
Tank and go to the seed opener, though. It's an old New Holland code.
And this cart too go to the seed opener because I switched the hoses. But yeah, I mean, that's what it comes down to, is understanding your equipment and the limitations that it can. And that's going to determine what types of questions you can answer. But absolutely good point. So here, maybe give him the red pen. Just randomly put 1 to 6 in different order here. So I'll go 6, 4, 2, 1. Can't see. I'm missing a number, aren't I?
1, you gotta do the outsides. 1, 2, 3, 4, 5, 6. I didn't do right the outside, sorry.
Oh, he's got 6 lines and you only have 4?
Well,
Huh?
I think he just
I just
added
drew
those.
another one in right now.
Oh, well, I can do that too. I can do that too. Okay. Now what am I missing?
One on that end, on that end.
Here?
Leave
Here?
one more there.
It's really hard to see today. Okay, Brian, you can put one in right there too. So in like an SMS software package, this is a little bit more complicated because you have to have a prescription map for each treatment. So if you have a tank, 2 different tanks, you need to have a prescription for each one of them. So all it means is you create one map for tank 1 and one map for tank 2. If this was just 6 different rates, it can all be on one map. And then you implement it and it goes. So this is now a fully randomized complete block design and gives you statistical power. So that means at the end of the day, you'll be able to analyze this and know without almost a doubt, you know, 19, usually you decide ahead of time what confidence level you want. We tend to use that 95% confidence level. That means 19 times out of 20, same result's gonna happen. You don't necessarily have to use that level. You could say 90%. So 9 out of 10 times I'm confident this is gonna happen. So let's give me a pen here. So here's a potential. This might actually get us into precision ag here, or variable rate. I'm gonna guess, we analyze our results. In this situation, cultivar Y at the high rate becomes our optimum level. You never know. Here, we're going to say cultivar Y actually just tends to be the best at our normal level. That means that we were at our optimum economic level, whether you knew it or not. Let's say this was the same thing. So this is our results, Y and N. So now that's good. We could probably jump to some assumptions that we can treat the rest of the field with that Y9. And it's pretty obvious now, if you want to, once you've built a prescription map and you have one variety now in your tank, it's very simple to build a prescription. And we say, okay, I'm going to seed the farm at this rate. The seeder's going to automatically adjust our rate in this area. And that's kind of the ins and out of it, right? Problem is, one of the beauties of this design too, though, is if you don't want to get complicated and have all of these 3 replicates, each block ends up being its own mini little research trial. You could take that block, put it on another field. Maybe now you want to discover more on the salinity side of things. You can take this block here, put it in another salinity zone and another salinity zone, and you can still run those statistics and find out whether you're seeing the same thing. So take a look through the booklet. There's some other designs we've done. Well, maybe what we'll do is kind of sneak in on the rest of the groups and see some different designs based on different design field. The problem is we can't come up with a cookie cutter approach for a variable problem. There has to be that variable approach to dealing with variable problems. And I think that's something that's been going wrong so far generally, you know, in precision agriculture in general. So, you guys have any questions?